Hello, everyone. My name is Anna Greca. I'm an associate professor at Harvard Medical School, a physician at Brigham and Women's Hospital, and an institute member at the Broad Institute of MIT and Harvard. And it's my distinct pleasure today to introduce the first author of this remarkable study in the JCI, who will take you through all of our findings and explain their implications, both for rare as well as more prevalent diseases, mitochondrial cytopathies. The first author of this study is Irini Heidi Sidhom. She is a remarkable MD PhD student in my lab, truly a rising star in the physician-scientist world. And she has really spent several years in the lab working on this project very diligently and with great success. So it's my great pleasure to introduce her and let her take you through our story.
Thank you, Anna, for that kind introduction. I am excited to tell you all today about how our lab is studying the mechanisms of rare genetically defined kidney diseases in the hopes of expediting therapeutic development in an area of great unmet need. Kidney diseases are a worldwide epidemic. With a prevalence of about 10%, approximately 850 million people worldwide have kidney diseases, including approximately 30 million in the United States. Furthermore, kidney diseases are a disproportionately expensive problem, with the cost of care in the United States exceeding $100 billion in 2016. But why is caring for patients with kidney diseases so expensive? First, there are no cures for most causes of kidney diseases. There are also few therapeutic options designed to treat the earlier stages of chronic kidney disease and prevent progression. And further, most people present in its most severe form, kidney failure, at which point the only two options are dialysis and transplant. And yet, nephrology lags behind most other fields of internal medicine in rates of randomized controlled trials. Common causes of kidney diseases are frequently multifactorial in nature and the downstream sequelae of systemic diseases. The mechanisms are poorly understood, making targeted therapeutic development difficult. Our lab believes two key principles can help to expedite therapeutic development for patients with kidney diseases. First, drug repurposing to identify novel uses for approved or investigational drugs has the capacity to reduce time and cost for the approval of new treatments for kidney diseases. Our lab has used screening of drug repurposing libraries to identify compounds like GDC 0879, which was initially developed as an inhibitor of mutant BRAF for the treatment of melanoma, and which we found to be podocyte protective in vitro. Second, our lab believes that studying rare genetically defined kidney diseases can provide important biological insights for the development of targeted therapeutics for the treatment of more prevalent diseases. Prior work in our lab has identified TRPC5 as a therapeutic target for the treatment of kidney diseases of the podocyte, a specialized filtering cell, and TMED9 as a target for diseases caused by toxic proteinopathies. Mitochondrial diseases are another heterogeneous group of rare diseases. Mitochondrial mutations leading to mitochondrial cytopathies are defects in oxidative phosphorylation. Thus, they clinically affect tissues with a high energy requirement. Unsurprisingly, the kidney, which has the second highest mitochondrial content and oxygen consumption in the body, is frequently affected in mitochondrial cytopathies. The functional unit of the kidney, the nephron, can be divided into its filtering unit, the glomerulus, of which podocytes are the specialized filtering cells, and the tubular compartment, tasked with refining the filtrate to maintain physiologic homeostasis through reabsorption and secretion. Of these processes, filtration at the glomerulus is a passive process, while tubular secretion and reabsorption is ATP dependent. Consistent with this, most mitochondrial cytopathies manifest as tubular dysfunction. However, a long-standing unsolved mystery has been that patients with mitochondrial mutations in the CoQ biosynthesis pathway do not have primary tubular epithelial injury. Instead, these patients present with podocyte dysfunction that causes steroid-resistant nephrotic syndrome, or SRNS, a disease characterized by kidney filter damage and proteinuria. CoQ is a ubiquitous membrane lipid present in all cell membranes. CoQ biosynthesis is mediated through a multi-protein complex. 5 enzymes in this biosynthetic pathway have been found to be mutated in autosomal recessive nephrotic syndrome. Mutations in these enzymes are thought to be loss of function, as patients have deficiency of the end metabolite CoQ. For this manuscript, we focused on an enzyme involved in the first committed step of CoQ biosynthesis, PDSS2. Using the model of PDSS2 dysfunction, we hypothesized that CoQ deficiency leads to podocyte injury and ultimately kidney failure through some cell-specific vulnerability. We investigated this pathway by first identifying druggable podocyte-specific disease pathways, and second, characterize disease-induced metabolic changes. To identify disease pathways, we perform single nucleus RNA sequencing on the kidneys from mice with whole body CoQ deficiency due to a spontaneous missense mutation in PDSS2. This model allowed us to compare perturbations in podocytes compared to tubular cells, such as the proximal tubule. Pathway analysis revealed podocyte-specific perturbations in expression of BRAF/MAP kinase pathway genes in CoQ-deficient mice. Treatment of CoQ-deficient mice with the BRAF-targeted compound GDC0879 reversed podocyte foot process effacement, the hallmark of podocyte injury. To dive deeper into the mechanism of injury, we used an in vitro model of shRNA knockdown of PDSS2 in podocytes to characterize metabolic changes in CoQ deficiency. Performing metabolomics and lipidomics, we noted that PDSS2 deficient podocytes had elevated levels of polyunsaturated fatty acids, or PUFAs, such as arachidonic acid, compared to scrambled controls. PDSS2 deficient podocytes were also more susceptible to cell death from arachidonic acid. Further, we found that arachidonic acid induced downstream activation of the BRAF/MAP kinase pathway, which was reversed by treatment with GDC0879. Returning to our mouse model, we observed that CoQ deficient podocytes upregulated GPX4, an enzyme that protects against PUFA-mediated lipid peroxidation. Further, GDC0879 restored GPX4 protein levels in diseased mice. In summary, we hypothesized that the study of a rare disease, such as CoQ deficiency, would identify druggable podocyte-specific vulnerabilities. Through single nucleus RNA sequencing, we identified podocyte-specific perturbations in BRAF/MAP kinase gene expression in the setting of disease. In vitro mechanistic work revealed alterations in polyunsaturated fatty acid metabolism, and that CoQ-deficient podocytes were more vulnerable to PUFA-mediated cell death. And finally, therapeutic targeting of BRAF with GDC0879 led to normalization of PUFA-induced BRAF/MAP kinase activation and rescued podocyte injury in vivo. Finally, using the publicly available NephroSeq database, we suggest generalizability of these pathways. Gene expression of GPX4 and RAF1 correlated with disease severity as measured by proteinuria in kidney biopsies from patients with podocytopathies. Therefore, in this manuscript, we demonstrate a model for the discovery of mechanism-based therapeutics for the treatment of kidney diseases, in which the study of rare diseases can provide insights into the molecular mechanisms of more prevalent diseases. Through the discovery of associated biomarkers, patients can be stratified based on the molecular characteristics of their disease to allow for repurposing of targeted therapies. With that, I would like to thank Anna Greca for her tremendous mentorship and guidance, as well as many other members of the Greca Lab for their contributions to this project. I would also like to thank our collaborators, including the labs of Katerina Quincy, Astrid Wines, Fei Chen, and the Broad Metabolomics platform, our funding sources, and the editorial board and reviewers at JCI for allowing us to share our work. Thank you.